hope people and examples presented. If you gather all together, which we usually know it's a very difficult task, gathering everyone together in your class is a good example of that.
Yes.
They cannot create a single fly, and if the fly takes away something from them, they cannot take it back. Yesterday I gave an example of malaria. Today I'm going to explain to you a case, which is my dad. So a fly stung him. Within it, it had a bacteria. That bacteria produces a protein, which we will learn today about it. It's called superantigen. And that's protein, literally what it done to him. So he had an infection because of that. So his lung was fully infected. He had ARDS, he was so close to use ventilator for breathing. And he was completely healthy before. Infection went up to the brain, stroke. So hearing loss from one ear, the other ear, 30-40% hearing. Kidneys went down, so he was so close to go under, I think, no, he went under dialysis twice.
Did they put him under coma or?
No, it was close to that. What else he had? Oh yeah, so the infection came onto his aortic valve, started growing there, vegetated and everything, so they had to do open heart surgery, valve replacement, they put a mechanical valve. So he reached a stage, he was 75 days in hospital, he reached a stage that, as I said yesterday, 30% of the people would only survive. Alhamdulillah, he was amongst that 30%, but his health is not going to go back, just because of that single thing. Okay? And today we're going to learn about what was behind that. Okay? And the key thing is, we do not acknowledge God, Allah subhanahu wa ta'ala, the way we should. So we should not become arrogant, I am doctor, this. We cannot even create sins. So today I want to be talking about antibodies and antigens. Usually this lecture was supposed to be a 4-hour lecture, but now we have summarized it and put it into 2 hours. So maybe less of group activities. Again, my email is this. You'll send me email and I'll send you send me all of them, yeah? So I'll send you all the slides over there. You can share it with us. Right, so hopefully by the end of the session, you'll have a good understanding about antigen binding molecules, about antigen, we talk about epitope, immunogenicity, antibodies, structure, function, and molecular antibody. This is the outline of the class, and let's go and talk about this. Why antigens are important? Why do you think antigens are important?
Because they protect our body. Because they protect our body.
Antigen protect our body itself? Huh? No. You're close, but no.
Antigen meaning antibody generated, right?
Yes, antigen means something that we have an antibody against.
And the antibodies are the ones that are against antigen.
Yes.
And they connect to the antigen.
So antigen can be anything on any molecule. Okay, whether bacteria, whether virus, whether our own cells. Okay, so this is the antigen. Why antigens are important?
Antigen? It's a very hard question. Like, if you ask why antibodies are important, I could answer.
So I said why antigens are important because they're important because we are able to, with them, we are able to detect them. So for detection. Yes?
Yes.
On top of detection, after detection, a good response against it. Whether immunity or immune regulation. Because if it's self-antigen, we want immune regulation. If it's pathogen, bacterial, virus antigen, that is not as toxic, we want immunity. It's immune system fighting against it. Okay? So antigens are important for that. So just like identifications that we have. How many sets of identifications do you have? Any people in the world has?
As a passport,
Well done.
ID.
Yes,
ID
so
card.
2. One for internal, domestic, Yeah, national ID cards, or sometimes we have in Iran, we call this Shenasname. Okay, we have the card and we have that as well. So that's one for domestic. Can I go with that Shenasname to Iraq? No. I go, what is this? Yeah. But for outside one, what do we have?
Your
Passport. Everyone recognizes passport. Okay, all countries. Similarly, ourselves, we have two kind of IDs. One for intracellular, so one for extracellular. Okay? And then, so imagine with this, the booklet that covers this, so all the printed information, let's say it's the antigen, which identifies my ID with someone else's. So the typings on it, let's say that's the antigen. The cover of the book, yeah, the book itself, similar we have in our immune system, is something called MHC, major histocompatibility complex, which all it does, it's a glycoprotein, and the antigen sits in the middle of it. Yeah? So it presents antigen. So, do you remember yesterday when I said, for instance, dendritic cells trapping the antigen, foreign antigen, where do they go next? Let's say I have a skin cut. Yeah, like that video I showed you of the mouse feet. It has an infection. What happens? Dendritic cells come grab a bit of that antigen. Where do they go next?
To the CD4.
Which organ is that?
Organ?
Huh?
CD4?
No,
Yes.
CD4 T cells. They give signal to CD4 T cells, but where does this happen? What organ?
Organ lymph node.
Spleen?
Lymph node?
Well done, lymph node. They go to the lymph node, they present to T cells and B cells and say, okay, which one of you can recognize? A B cell, sorry, a B cell would come and say, okay, I can recognize you. Once it can recognize, it gets the signals, then what will happen? From the same B cell with the same structure receptor, which we call it B cell receptor, BCR, we have clonal expansion. So same cell would be expanded in loads. Why? Because it wants to go and fight the enemy. So we need more of that specific B cell. Whereas these ones, the receptor would not bind. Okay? So therefore, antigens are very, very important because with the antigens, we are able to detect and then show the response. Right, so in this video, you can see the process of immunity. So this shows, let's say, a macrophage. We have pathogens, and it engulfs a pathogen in a phagosome, digests it, we put it on those molecules I told you, MHC. So antigen is presented on that glycoprotein. And now this cell, it will be classed as antigen presented cell because it presents a foreign antigen. And what happens, CD4 T helper cells with the right receptor comes, and if it attaches and gets a signal, these pass the signal to them, so they become activated. And one thing they do first, actually, clonal expansion. From same cell with the right receptor, we have loads. And then this sends signals, in brackets signals are called cytokine, to other cells, such as cytotoxic T cells, B cells. And what happens once they become activated with T helper cells, which express
CD4.
Well done. And once cytotoxic T cells and B cells get activated, let's say this is the cardiomyocyte, virus is inside, the antigen inside of the cytoplasm is presented on the MHC molecule number 1, which is intracellular. And cytotoxic T cell receptor, its receptor recognizes that antigen, which is from internal, and then it tells him that you have a virus, you have to die, and that cell dies, because it has a virus inside. Okay. B cells, what happened? The B cells with the right B cell receptor that can recognize the antigen, they get the signal, so again, clonal expansion, and also they differentiate into plasma cells. Plasma cells, what do they do? Secrete that specific antibody that we have into the, from the lymph nodes, into the circulatory and everything, lymphatics, and then they connect to the pathogen, and literally ensuring that the macrophages comes and engulfs them. And as we mentioned yesterday, adaptive has memory. So some of these cells become memory B cells, some of the CD4 T helper cells becomes memory T cells, cytotoxic T cells also become memory cells. So this is the process of immunity, right? I mentioned MHC1, just let me mention MHC number 2 as well. Remember I said we have two MHC molecules, right? One is for intracellular antigens, such as, let's say, viruses inside. Yeah, virus is not like in a phagosome because they're tiny. It go inside, integrate itself into inside. And anything intracellular, like domestic IDs, they're presented on the molecule MHC number 1. Okay? Don't worry about the MHC structure and function and everything. You have a 2-hour lecture on this. So MHC number 1 is for intracellular antigens. MHC number 2 is for extracellular antigen. Extracellular antigen like what? You remember yesterday I talked about macrophages?
Yes.
Yeah?
Yeah.
That it was the bacteria was outside, puts it in a bubble or a phagosome and then brings it inside the cell. And then digests it, kills it. Then it puts it on the MHC number 2. Because this was outside, it was not inside.
The
Yes.
broken bits would be put on.
The broken bits would be put in on MHC number 2, and MHC number 2 will come on the membrane of the macrophage, and the macrophage therefore will be sort of an antigen presenting cell. What time is it?
It's 8 to
12 or 10? Let's say 12 for 2 minutes. The rest will appear in the other. Name, student number, and date. Right. Any questions?
Sorry.
Right. Let's summarize innate and adaptive. Remember what were the cells of innate we talked yesterday? Dendritic cells or DC, yes.
Macrophages.
Macrophages, which used to be monocyte, yes, activated those big
natural killers.
natural killer cells, yes. Those big phagocytic cells,
Eosine,
eosinophils, well done,
basophils,
basophils,
and neutrophils.
neutrophils, and other components, physical barrier, epithelial barrier, complement, which you mentioned. Okay, I'll have one slide on complement today. Points regarding adaptive immunity. What cells did you say? Yeah. Remember what marker, CD marker, B lymphocytes express?
B is from bone marrow,
Yes.
T from
Thymus.
thyroid,
And you know the marker, CD what?
CD,
CD, what's cluster of differentiation, remember? CD number. B cells express what marker?
CD4 or 8?
Is on T helper cells.
CD4?
CD4 on T helper cells. CD8 on
The cytotox.
cytotoxic, CD19. Next one. Right. Adaptive immunity is much more specific, if you remember. Diverse response. The first sort of response it has usually takes longer. The second time it encounters antigen is much faster. And what else? It has memory, if you remember. Okay, from my lecture and Dr. Nuraj. So, whereas in innate, it has a specific response, but it's not as specific as that. Okay? So therefore, antigens are more important in adaptive. Okay? And we're going to be talking about that. So literally, this table is summing up what we discussed. And likewise here. So this is I think from Janeway, this text is from Abalabos. Right, so the cells inside the adaptive, we have B lymphocytes, which one of the role is expressing the antibody once they differentiate. We have helper T cells. Their role is, if you remember, activating the whole immune system. Cytotoxic T lymphocytes, targeting the cells which are infected with viruses, and they kill them directly. Okay? Therefore, the T cell receptor on cytotoxic T lymphocytes recognizes antigen presented on MHC1 or 2.
2.
2. 2 is for extracellular or intracellular.
Extracellular.
Well done. So, virus is extracellular antigen or intracellular antigen? Is virus outside and then does it integrate inside the cytoplasm?
No.
Once it's inside the cell, where does it go?
It's taken out.
From outside to inside by the receptor.
Yes, so let's say coronavirus. Coronavirus, does it replicate outside our cells?
No, inside.
Inside the cell.
Inside of.
So if it's inside the cells, the antigen of the corona is presented on MHC number 1 or 2?
One.
One.
One, because it's intracellular.
Yeah.
Cytotoxic T cell receptor recognizes the antigen presented on MHC1 because it's intracellular. Why? Because the T cell, cytotoxic T cell, which expresses CD8, it sees a cell, it has intracellular foreign antigen. And it can recognize it, it orders to die. Yeah? Whereas these cells, the T cell receptor of CD4 T helper cells, they recognize antigen on MHC number 2, which is foreign. Okay? So if a molecule is outside, they have to be alerted. Why? Because then they alert other cells. Because if a pathogen is outside there, alive, that means definitely should be sometimes, it can be inside our cells as well. So if they get activated, they can activate other cells. Okay? And their T cell receptor recognizes antigen on MHC number 2, which is extracellular. Okay? Actually, while on that, let me. Yeah. Let's talk on antigen presentation and then I'll go back. So, any cell that so I ask you this question. Which cell of us do you think should express MHC1? MHC number 1 is for intracellular antigen. Okay? Which cells of us do you think it should have MHC number 1?
The first one.
Which first one? Here? Or I ask you another question. You said we have identification 2 types. One for internal, like national cards you have, okay? One passport for external. Who does need national cards?
And I think the skin.
No,
The blood.
Syphilis?
Huh?
The people inside the country.
Okay.
They need
Which
an
ones?
identification.
Just the pilots need them?
No.
Who needs them?
Everyone.
Everyone. MHC number 1 for intracellular antigen, all cells have it. All cells. Except red blood cells. Okay? Why? Because all cells should always show that, listen, whatever is inside me, it's fine, it's safe. Just like all of us. Which country are you from?
Iraq.
You're from Iraq. Can you live in Iraq without ID? No.
No.
And get services from the country? No. Yeah? With national ID.
Yeah.
But does in Iraq or Iran or Nigeria or Pakistan, does everyone have passports?
No.
No. Whoever needs to fly out, you guys, you came outside, you need passport.
Yeah.
Prior to that, if you didn't travel, you didn't need passport.
Of course.
So all cells, MHC number 2 for extracellular, okay? All cells do not express MHC number 2.
So the red blood cells, and that's because it's removed all the time.
The red blood cells do not have MHC1 because they don't need, literally, they're kind of semi-dead cells, and their role is something physiological.
That's
Okay.
because the reason because
And it's short-lived.
It's moved all the time.
And it's literally short-lived. Okay? And it's literally without nucleus or anything. It can't do much. Okay? So literally it just carries oxygen and that's it. Dead.
What about blood cells?
Other blood cells, yes, of course. All of them should. All cells will
Well,
have,
I mean, why?
have MHC number 1,
Okay.
which is for intracellular. But MHC number 2, which is for extracellular antigens, only antigen presenting cells have it. Like which ones? Dendritic cells, macrophages, and B cells. They are professional antigen presenting cells. I don't know in your country, but in some countries you have 2 types of passports. One is like 30-page passport, the other one is 50-page passport. 50 pages passport are for those who travel a lot.
Yeah.
You pay a bit extra, so these are the 50 pages passport. They always express MHC number 2 for extracellular high amounts. Okay? They can go higher amounts as well, but always high. Then non-professional antigen presenting cells such as fibroblasts, microglia, different locations, different places. Based on the role, they highly express, then downregulate MHC number 2. Okay? So therefore we call them non-professional antigen presenting cells. This is the list I told you. And you saw this video yesterday. Role of antigen presenting cells, dendritic cells. These green cells are dendritic cells. There's infection here, and as you can see inside the mouse feet, they all go and migrate towards that direction. Okay? So, this is for antigen presentation. Let's go back to here. Right, so now let's talk about molecules that bind to the antigen. Again, they're quite important.
So we'll enter on the inside of the cell.
I'll talk. So, we said antigens are very important. Why? Because for detection and later on for response. Yes? So this is why it's important. In which immune system we said there is more diverse and specific responses? In adaptive. Therefore, we're in adaptive. We're talking about antigens in adaptive. And what molecules do we have? So we can have a B cell receptor, which is membrane bound. It's attached on the membrane, cell membrane. But if it's soluble, we call it referred to as antibody. So they can attach to an antigen, as well as T cell receptor, which is membrane bound. Okay. Or the other molecule which we said it binds to antigen is what? We said the B cell receptor or antibody, T cell receptor. What's the other molecule? I said it that is presents the antigen.
MH2.
MHC. Well done. Whether one or two, both of them. Yes, MHC 1 or 2. They also bind to antigen. In order for antibody or B cell receptor to attach to antigen, we only need two molecules. One is the B cell receptor or antibody, and the other molecule is the antigen. Yeah, that's it. But for the other one, for T cell receptor, we need three molecules. We need a T cell with T cell receptor that recognizes the antigen which is presented by MHC molecule, which is on an antigen presenting cell. Okay? And that MHC can be MHC number 1 or MHC number 2. So for T cell binding, we need three molecules. Yeah? Therefore, the antigens that T cell receptor can recognize, okay, they all should be bound by the MHC. Yes? T cell receptor, can they recognize that? Let's say this is an antigen on its own. T cell receptor can recognize that? No. Why? This should go and be presented on an MHC molecule on an antigen presenting cell. T cell receptor recognizes this presented by MHC. Okay? So therefore, T cells cannot detect soluble antigens unless they're presented by MHC. Okay? Whereas antibody or B cells, yes, they can. Because they need only two molecules, B cell receptor, and they need to attach to the antigen. Yeah? And next thing, what type of molecules or antigens they recognize? B cells, they can recognize everything. Protein, polysaccharides, lipids, anything. Yeah? Whereas T cells can only recognize the molecule which is presented on the MHC. Yeah? Which the structure of MHC, something like this, let's say. Okay? And what molecules can attach there? Linear peptide. So we cannot have a big protein start sitting there because it doesn't fit. Only linear peptide, and it's around 15 to 20 something to 30 something peptide that is presented on the MHC, and T cell receptor can recognize that. Right, and how antigens come and sit here or on the T cell receptor presented by MHC, how the molecules interact with each other. You know, like two magnets, if you have the two magnets on the opposite poles, they attach to each other. Are they linked to each other always? No.
They make sequence.
So literally the two magnets that we have, the opposite poles, we put them, they stick to each other. Is this like metal, I don't know, sticking and you cannot separate them anymore? No, you can dissociate them. Yeah? Antigen and antibody interaction, or T cell receptor or MHC, is exactly like this. It's not covalent bonds that they stick there forever. They're very strong bondings, but it's reversible, so it can be dissociated. Okay? Antigen and antibody can be dissociated, just like the two magnets. And what bonds are important? Van der Waals, hydrogen bonds, hydrophobic, hydrophilic properties of the molecules that we have on the B cell receptor or antibody and the antigen. It makes them to stick to each other. Okay? So bear that in mind. Right. Actually, this is what we mentioned on the previous slide as well. But here is showing MHC. In the next lecture, we'll be talking about affinity, so don't worry about it.
Doctor, for the exam, what should we know about this figure? For the exam.
Listen to the lecture.
Okay.
Okay, at the end you'll understand the whole thing. I said it in the beginning, a brief understanding about what stuff
It's the first time to me.
Yeah. So antigens. Any substance that can be specifically recognized and immune response against it, we regard it as an generated immune response, we can say antigen. And so B cells with B cell receptor, T cell with T cell receptor. I mentioned what type of molecules we can recognize, and TCR or T cell receptor can recognize what molecule?
Peptides.
Peptides. Why?
Because they have to be small.
Why?
So
Because they have to be linear.
Huh?
It can.
This doesn't path like this.
Yes, well done. So because only small molecule, a linear peptide can fit in the MHC,
Yeah.
not a big protein. Yeah?
Yeah.
Right. So what are antigen presenting cells? We mentioned that. Let's go to the next slide. So one key thing for antibody and antigen interaction, this blue part is showing the antibody side. The red are showing the antigen. Okay? So it can be antibody, it can be T cell receptor, it can be MHC molecule. But the key thing to bear in mind is that the interaction, as we mentioned, is supposed to be fitting each other. Okay? It's like the glove and the hand model, lock and key. Yeah? So they are complementary to each other. Yeah? And if they are not, if the interaction is weak, they'll have no attachment and no further responses. Yeah? So how B cell receptor can recognize it? If it has the right receptor for this, it can be bound and show the signal. But if it doesn't have the right receptor for this, it will go away. Why? Because they need to have the 3D structure and the elements of the molecules that we have, that they fit into each other, is important as well. So this is showing the antigen, for instance. It should fit in that. And the molecules here and on the antigen are very important, because it's with these molecules that we have hydrogen bonds between antigen, between antibody, yeah, or T cell receptor or MHC. And they have hydrogen bonding, so they have a reversible reaction, and they will be stuck to each other with high specificity, and then later on they'll be dissociated. Right, so this is showing an antibody, okay, and this is showing an antigen. Epitope is the location where the antibody recognizes on the antigen. So imagine my mobile phone here. Yeah? Let's say this is a big protein, and so it's antigen. Let's say antigen X. Okay? How many sites or how many locations antibody can come and sit and recognize? One, or just here? What about here? Different structures?
This is the front and that is
Yeah, so it can have many. One antibody can sit here, one antibody can sit here, one antibody can sit here.
That means we have
So meaning this protein X has many epitopes. Yeah, many epitopes. Yeah? Or the other name for epitope is antigenic determinant. If it shows you well like this. Yeah? So epitope or antigenic determinant on the antigen molecule, which means recognized by the B cell receptor, antibody, T cell receptor, or MHC. And to this region here on the antibody, we call it paratope. Okay? So paratope on the antibody, or T cell receptor, or MHC, and epitope on the antigen. How many epitopes this phone has, for instance? Many. How many this table will have? Many.
Many.
Why? Because the structure is different, and different antibodies can come and sit different locations. Right. How many types of epitopes do we have? Okay. 2. Anyone has a hands-free? Everyone has Bluetooth. Anyone has hands-free? Do you remember the structure of the proteins? Primary sequence, secondary, blah blah. Yeah? Yeah or no?
Maybe.
Huh? Maybe. So a protein, how many structures does it have? So they say primary sequence, which is what?
Secondary,
The peptides.
and the
Secondary,
yeah.
yeah.
We have 4.
Tertiary, quaternary, yes?
Yeah.
So this is a protein, yeah? And primary sequence. Now it turns for, let's say, tertiary structure.
Will be purple.
If an antibody comes and attaches to this sequence here,
Yeah.
if I open it up, these are all in a linear form, yeah? So the antibody recognizes peptides on this. So to this epitope we say linear or sequential. It's in a sequence. Yeah? But let's go back to this. But if an antibody recognizes here, if I open up
What's that?
2 different locations in 3D, they came closer to each other. To this we say conformational, because in the conformation, in the structure, the amino acids came to each other, and the antibody can recognize that. So we say conformational or non-linear or sequential. And you can see it even better on this slide here. So linear epitope or sequential. But here, recognizing these 2, this would be conformational. Okay?
Which one is more activity?
Which was more active?
Yeah.
Doesn't really matter, no, nothing. But I ask you a better question. T cells can recognize which one?
Cells?
Linear. Yeah, why?
Because it's small and
Small
light.
and fast.
Because
These are
MHC2
Yeah.
can be MHC1 or 2 only can have linear peptides. Sequential, no. So my next question: does T cells can recognize a big protein?
No.
Huh?
T cell?
T cells
No,
can
T
they
cell
recognize a big protein?
no, small.
So the better answer is it can recognize a big protein if it's breaking up into linear peptides. Yes? If a big protein is internalized by cells, breaking up into linear peptides presented by MHC 1 or 2, then T cells can recognize. Yeah? Otherwise, big protein on its own, as we said, no, we cannot. Yeah? So this is linear and conformational epitope. So, protein X, what does this mean? Linear epitope, as we mentioned before, this glass here is all one sequence. So this is linear. But between the glass and the case and the phone itself, 3 different materials getting closer to each other, this is conformational. But another epitope we can have, okay, it's called neoantigenic determinant. This protein, once it's internalized, it might be phosphorylated, it might be literally certain post-translational modification could happen to it. And for instance, it can be degraded like this, and it has a new shape. Okay? So therefore, it will have a new set of epitopes. Okay? So methylation, for instance, adding a CH3 to it, it changes the structure. To this we call neoantigenic determinant, meaning a new epitope has been made. Okay? And through these post-translational modifications, which again on this slide it has summarized all three. One, conformational, so in the 3D structure they all came close to each other. Linear, one antibody recognizing that sequence, the other one antibody recognizing that blue sequence. This is linear. Let's say on this case we have an enzyme which cleavages on that side, so it breaks up the protein. Now we have new set of epitopes, and therefore it's called neoantigenic determinant. Yeah? Any questions?
So we have 3 epitope types.
3 epitope types. Linear, conformational, neoantigenic determinant. Right. 2 definitions you need to learn. One is immunogen. Immunogen means any antigen which immune response is initiated against it. So we have immunity against that antigen. Okay? Tolerogen means if the antigen is taken in a way that our body shows tolerance towards it. Okay? So, part of the immune system. Yeah, do you remember we said yesterday, the immune system does 2 things. Fights the bad guys. When the bad guys are gone, calm down. Immune regulation or tolerance. Okay? So if an antigen is administered in a way that we show tolerance to it, we do not show immunity, we call it tolerogen. Like how? Eating food. When we eat food, it's all foreign molecules. Yeah, we eat meat, vegetables, everything's foreign antigen. But the way we take it, we show tolerance towards it. How? We have 2-hour lecture on that, in the tolerance. Okay? Right, let's talk about T-cell receptors. So, big protein can be recognized by T-cell receptor? Yes or no? Big protein.
Yes.
By T cell?
By T-cell.
No,
Well done. Why not?
because it's so big.
It's so big and it needs to be
just like
digested into linear peptides presented by MHC. Now T-cell recognizes it. One question. Does T-cells can recognize linear peptide on its own?
yes, just like
No.
No. It should be presented by MHC. Yeah? So my question was, can a T-cell receptor recognizes this hanging around somewhere? No.
No.
That should be presented by MHC molecule. T-cell receptor recognizes the antigen and recognizes the MHC.
But for a B cell, it would.
Well done. B cell can recognize this, and it can recognize this would be linear. This in the middle would be conformational, one sequence from there, one sequence from there, one sequence from there. Or if you have new antigens formed, you can have that. Right. What was the role of CD4 T helper cell? Remember? CD4 T helper cell. What was its role? You remember? You just record, yeah? You say it's in there. My phone memory remembers it. So, the role of CD4 T cells. Remember I said if there's going to be a bombing or something, then central siren goes around. Everyone gets alerted. Everyone gets activated. We as non-military, we start going to the bases, to the refuge sites. Okay? The military, they go to the military bases. Yeah? CD4 T helper cell kind of do this with the immune system. Literally alerting and activating the immune system. Right, my question to you is that button, let's say this red button, this orange button is the button. If you press it, the central siren will go inside the Kish Island. Yeah? Will we be putting it easy there so everyone comes and touches it?
No.
It's supposed to be kept safe with the protocol, this. So in order for T cells to become activated, yeah, CD4 T helper cells, in order to become activated, they need 3 signals, not just 1. Before you remember, I said if T cell receptor recognizes the antigen presented by MHC, the T cell will get the order clonal expansion of itself and then show its function. But this is just 1 signal. It needs 2 more signals. It needs 3 signals. Second signal that it needs So CD4 T cells having T cell receptor recognizes the antigen presented by MHC number 2 on an antigen presenting cell. So this is signal 1. The other signal it needs is these molecules, co-stimulatory molecules we say, okay, which act like this. So T cell, let's say T cell receptor recognizes the antigen presented by MHC. Co-stimulatory molecule is like this. It makes this interaction to be stable, kept. Okay, so this is signal number 2. Signal number 3, CD4 T cells required before to expand, is inflammatory cytokines. It's danger signals literally around it, which means there is inflammation, there is a bacteria attack, there is something. So if CD4 T cells have this signal, then they will be activated. And once they activate it, they alert the whole immune system. Okay, so this is why antigen are very important. Without antigen, okay, and no TCR binding, even if there's co-stimulation or inflammatory cytokine, this cell will not be activated. But if we have antigen and co-stimulation and danger signals or inflammatory cytokines, then they will be expanded. Okay? So mitogens, what are mitogens? They can be parts of bacteria, okay, or sometimes even you can have it in vegetables. Sometimes we have molecules which act as a mitogen. Mitogen on itself, what does it mean? It means generates mitosis. Remember mitosis, cell division?
Yes.
Mitogen means it generates mitosis. What does it do? It's literally a molecule that sometimes bacteria have, or sometimes we have in food. We have structures which act as a mitogen in our food and veg. That what happens, for the bacteria at least, it resembles co-stimulation. Okay? So come and sit on the CD4s, and CD4s thinks that it has co-stimulatory signaling between T cell and antigen presenting cell. Okay? And what does this do? This will activate the CD4 T cells. It will get them closer. Out of the 3 signals, you remember? So let's say you have a T cell receptor. This is its receptor. Okay? It comes and tries to recognize the antigen. It wants to go away because it doesn't bind really strongly here. Okay? It wants to go away, but with the mitogen, it gives it adhesion, and it thinks there is a co-stimulation, signal number 2. With little bit cytokine from danger, wherever, okay, now this cell will be activated, CD4s. And bacteria produce this co-stimulation, or bacteria has this mitogen molecule, okay? And they bind to the CD2 on the T cell.
So mitogens are helpful for us.
You tell me that. So I said bacteria, such as like LPS is from bacteria. They make mitogen. Mitogen mimics co-stimulation for T cells, meaning it makes them easier to get activated. Yes? My question to you, I'll give you 1 minute to discuss U2, U2, U3, and U2 at the back. So how many groups do you have? 1, 2, 3, 4. 4 groups. My question to you, I said some bacteria have mitogen, meaning the mitogen, it mimics, it shows like it's co-stimulation for T cell. What does that mean? There's a higher chance T cell gets activated. Now, question to you, if T cells get activated, there's a higher chance we have immunity against bacteria. Yeah? Because it's switched on. But why bacteria wants the immune system to be turned on? Do you understand the question? Mitogen is made by bacteria.
After the adhesion?
It's made by bacteria. What does it do? It mimics adhesion or co-stimulation. Yeah? Co-stimulation makes T cells to be activated. Why a bacteria wants T cells to be activated? Discuss in your groups. You understood the question? Answer. You got answer?
Maybe.
You got answer? No.
Yes.
You?
Like Arya.
What about you two at the end?
We,
Who's sure about their answer?
no,
I'm not sure.
we're not sure.
Okay, who thinks this is the right answer?
It's made like
Maybe.
barrier between the bacteria and T cell, the antigen and T cell.
No, this mitogen, it actually,
It reduced
what
by.
does the mitogen do?
It's
Ah.
reduced by-
Listen,
It
mitogen, what does it
activates-
do? What does the mitogen do?
It activates.
It kind of activates,
Activates.
it gives the
Yeah.
co-stimulation, yeah?
Yes.
So co-stimulation is one of the signals we need for T cell to activate,
Mhm.
yeah? So what does,
I think some and the T cell substructure will help the bacteria or the antibody.
why T cell, if this T cell gets activated,
Because
The
it will
other
change
kill the
cells-
bacteria.
No, no, because the antigen will secrete in the mitogen, so it will change the structure of the T cell
yeah?
and will affect it on the cell. I think.
So yeah, but the T cell will become activated.
Of course.
More activated will come with the antigen. That will change the structure of from
Yeah.
help to help the antigen. From helper cells to help the antigen.
No, but if T cells get activated,
Yeah.
it means immunity,
It
means
activates.
higher chance of seeing the bacteria and killing the bacteria.
I think
Yes,
we'll
but
change
maybe-
the structure.
But I
That's
think
my-
it once
Maybe
you
the bacteria
destroy the
has,
whole
like
immune
enclosed
system
in
from the cell.
some capsule.
I think
by
what will
the-
happen.
Like 1%.
And by alerting the body would be confused, like too much chaos.
Too much here.
And so it will might start attacking the other healthy
Exactly.
ones.
That's
Cause
the-
like sepsis or
Yeah.
inflammation.
You're getting there as well,
Yeah.
okay? Because T cell receptor recognizes antigen. Strong
Yeah.
binding? No, this isn't strong binding. Yeah? It doesn't recognize it. And this is presented by MHC. Yeah? It wants to go away, but because the mitogen, hold both my hands, this is co-stimulation, the mitogen acts like this. I want to go away, but this gives, no, the signal is there, it's pretty strong. So it activates this cell, this T cell. And this cell, the receptor is for what? It could be for my self-antigen.
Yeah.
Once this cell is activated, it will come and start targeting myself. And what will happen is that do we have more self-antigen or more bacteria antigen in our system? Self. So the T cells go crazy, it starts coming attacking. What does the bacteria do? Starts living and expanding and everything. And at the same time, bacteria, this is the receptor for here. There's another receptor for my heart. The mitogen gives co-stimulation to that T cell as well. So therefore, it causes chaos, and then bacteria can go and divide and colonize and everything. Okay? And this is a list of mitogens. So sometimes, for instance, pokeweed or jack beans, red kidney beans, they can act as mitogen as well. That's why we have food allergy. When they say do not eat loads of broad beans, for instance. Yeah? You can have an allergic reaction or whatever. Sometimes you have allergy, but sometimes if you eat too much of them, it will be toxic. Yeah? It's because of this. Their structure is mimics, binds to the signals on T cells, so T cells think it's co-stimulation. Okay? Right, shall we go next? Now let's talk about super antigen, the thing that my dad got. So my dad was stung by a fly that I had Staphylococcus aureus, and that makes something called staphylococcal entotoxin, which is a super antigen. And what does that do? This does not bind to C as a co-stimulation. This one covalently links MHC to T cell receptor. Meaning, even if the antigen is not there for that the right T cell, the T cell thinks it's attached to it, and it cannot go away. And that super antigen, which is made by the bacteria, can attach to any T cell receptor, and what does it do? Suddenly the whole immune system starts fighting our own system. And you have multi-organ failure. Okay? What does it do? It covalently links one of the subunits to the T cell receptor of MHC. So T cell receptor comes and sees, doesn't recognize antigen, want to go away, but the super antigen, so MHC presents antigen, T cell receptor, they covalently linked now. Can't go away. T cells with little bit of co-stimulation molecules cytokine, these T cells get activated and start killing ourselves, fighting ourselves. Okay, so this is super antigen. Right, immunogenicity. What time is it? Okay, we're doing good with time. We'll talk about this and then you can go for break. Immunogenicity, meaning we have immune reaction, okay? So the more molecule is foreign towards me, will I have more immunogenic reaction or less immunogenic? If it's more foreign towards me. I'll give you an example. Who's your twin brother? Where is he?
He's sick.
He's sick. So imagine if they're identical twins. You give your kidney to him. Will his body be rejecting it?
No.
But let's say if we get a kidney from, let's say, an animal, we do xenotransplantation, will it reject it?
It will have more higher chance.
A very, very high chance. So the more foreign towards us, higher immune reaction or more immunogenicity. Okay? Size. The bigger the size, more immunogenic or no?
Yes.
Yes.
Why?
More active.
Because we can see it easier.
Yeah.
The immune system can see it easier. Generally, molecules should be bigger than 100 kilodalton in order for the immune system to recognize. Anything lower usually doesn't recognize. But we can train the immune system in a way that even can recognize molecules less than 10 kilodaltons. How? Those molecules, we call them haptens. Okay? What are haptens? Any small molecule that on its own the immune system cannot recognize. But if we stick, like for instance this is dinitrophenol, and there are two different isomers, yes? Immune system on its own cannot recognize this because it's a very tiny molecule. Okay? It's not a linear peptide. Linear peptide, compared to this room, if you want to say, it will be covering the whole room one round and maybe second round. Okay? This is how big a linear peptide will be compared to this. But we can train the immune system in a way that even shows immune reaction towards these two isomers differently. So we have one immune response for this isomer, one immune response for this isomer. How? This molecule on its own, which does not show any immune reaction, we call them hapten. But if we bind them, so let's say it's this red triangle, okay, that tiny molecule here is this red triangle. If we bind them to a big immunogenic protein, we covalently link them. So that hapten molecule is tiny, no immune reaction. Or we get a big protein, immunogenic protein, we covalently link them towards each other. Now this big complex, we inject into an animal. The animal will have immune response against the big protein, like that. It will have antibody against that. It will have immune response against this epitope here, recognizing both. And also it might have immune response against that, which is the little DNP molecule that we had here, where it's these molecules. Yeah? We can have an immune reaction against that. So therefore, to this we say hapten, tiny molecules on its own, they cannot be recognized by the immune system, but if we attach them to a bigger immunogenic protein, covalently attach them, link them, then we can have an immune reaction against them. Right, let's go back. So this is regarding size. Complexity of the molecules. From proteins because we have diverse elements in it, so more immunogenic. Whereas lipids, it's all CH2, 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 CH3, yeah? All similarly looking structures, less immunogenic, okay? And if an antigen is particles rather than soluble, there's more chance to be detected, okay, and more immunogenic. If my question to you, if we have an antigen or linear peptide, and if there is a strong interaction with the MHC, is that more immunogenic or less? If the linear peptide antigen is very strongly attached to the MHC compared to another linear peptide, which is attached weak, which one is more immunogenic? Strong or weak?
Strong,
Why?
because it will stay and will.
Well done, and therefore higher chance for T cell receptor
Yeah.
coming and recognizing. So therefore, this is what it's saying. We, each of us, our MHC molecules look different towards each other, okay? And if the antigen is bound more stronger to that, there's a higher chance we can show immune reaction against it. Therefore, that's why some people, when we go on, for instance, whether it's in type 1 diabetes, whether it's in autoimmune diseases, with a specific sequence of MHC, okay, there's a higher chance of some people getting certain diseases. Why? Because it shows that antigen higher, and there's a higher chance we have immune reaction against it. Route of administration, okay? Subcutaneous is the most immunogenic, intragastric is the least. You remember toleragen? We said if you take the antigen in a way, we show tolerance towards it. Okay, if we eat it, for instance, intragastric. So intragastric, the most least immunogenic, subcutaneous, the most, then IP, then IV. That's why all of our allergy tests, I don't know if you've done allergy tests. Anyone done allergy tests here? No? So what do? They get the antigen of different molecules and they put on your skin. And then they try to look if it's swollen or not. Why? Because the skin has loads of, it shows a much quicker immune response. Okay? Therefore, immunogenicity higher if injected subcutaneous. Dosage. If the dosage of antigen is very, 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 very little, what will happen?
Oh.
Huh? Do you have immunogenic reaction? No. If it's intermediate, yes. If it's very, 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 very high dose, do you have immunogenic?
Yes.
Very, 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 very high.
No.
Yes.
I'll give you an example. Let's say me and you, okay, we're friends. If I slap you like this, what will happen? Like this.
Nothing.
No.
You smile. No. But if I slap you intermediate like this, naturally just like that. Naturally you slap my face. I do it with this hand, you do it with the other hand. Yeah? Or if I double bang slap you, you go like, stop trying.
Yes.
Yeah? Immune system like that. Intermediate dose is the highest immunogenicity. If the dosage is very, very, very high, immune system will go kind of, it will show tolerance. Okay? Because like, what can we work? We want. Yeah? So this is what I discussed, and this is in the table. Size, the larger the size, higher immunogenicity. Dosage, intermediate, higher immunogenicity. Subcutaneous is the highest immunogenicity. The structure of the molecule, the more complex it is, like proteins, higher immunogenicity. The more it's different from our own proteins, the more foreignness it has, higher immunogenicity. And if its interaction with the MHC is higher, then higher immunogenicity. Yeah? Make sense? Mm-hmm. Right. Let's summarize and then we'll complete. So we talked about innate and adaptive. Antigen binding is much more important in adaptive. Then we talked about the molecules which bind to antigens, which is MHC. And we said MHC we have type 1 and type 2, yes. MHC type 1 for intracellular or extracellular? Intracellular. MHC type 2? Extracellular antigen. Yeah. And CD4 T helper cells recognizes MHC2 or 1?
2.
2, well done. Cytotoxic T cells?
1.
MHC1, which is intracellular, well done. Because if the intracellular antigen is foreign, cytotoxic T cell kills that cell. Yeah. Then we talked about epitope, the surface of the protein or antigen that antibody recognizes is called epitope. How many types of epitope we have? Or antigenic determinant? 3 we have. Linear or conformational, which is the structure brought them together, or neoantigenic determinants. And to the part on the antibody that recognizes the epitope, we say it's called paratope. Then we talked about T cell antigen binding, which we talked about, and presentation by MHC, co-stimulation, and we talked about mitogen and superantigen. Yes, we talked about immunogen, any antigen that immune response is against it, called immunogen. If it's we have tolerance towards it, like if we eat it, we call it tolerogen. And then we talked about immunogenicity, how we can have more immunogenic reactions. Yes. So this is this. That's what time is it? 9:02. Come back at 9:12.